Hi, my name is Dr. Yasser Osman, and I'm going to talk about gas chromatography. What is gas chromatography? Gas chromatography is an apparatus that has been considered the most accurate method to measure the concentration of inhaled anesthetic vapors. What is its clinical use? We use it to measure the volatile anesthetic agent for anesthetic concentration and in the operation room to detect the percent of pollution. Also, we can use it to detect other anesthetic drugs such as barbiturate, benzodiazepine, steroids, etc. It has the advantage of that it can detect very small trace elements of this substance. A bit of historical background. In 1901, a Russian scientist invented this principle by performing a research on the plant pigment, and he was able to separate the different plant pigment using such technique. How did he do that? He put a limestone in a column, then he put the pigment on top of this limestone. He left it. By time this extraction of the pigments from the plant separated into different pigments and different zones. Each zone has its different color, and hence he could extract the different pigments and its zones. He called this technique chromatography, which means a Greek word. Chroma means color and graphene writing in color. How does chromatography work? In all chromatographic separation, the sample is transported in a mobile phase. The mobile phase can be a gas or a liquid. The mobile phase is often forced through a stationary phase, held in a column or on a solid surface. The stationary phase needs to be something that does not react with the mobile phase or the sample. The sample then has the opportunity to interact with the stationary phase as it moves past it. Samples that interact greatly appear to move more slowly, while samples that interact weakly appear to move more quickly. Because of this difference in rate, the sample can be separated into their components. In a chromatography column, flowing gas or liquid continuously replacing saturated mobile phase and results in movement through the column. As a material travels through the column, it distributes between the stationary phase and the flowing mobile gas or liquid carrier phase. In a mixture, each component has a different distribution coefficient and thus spends a different amount of time absorbed on the solid phase instead of being carried along with the flowing. This is a slide that shows the separation phases of the chromatography. Here we inject the sample with its two components, the red and the blue one, and the flow of the mobile phase is continuously present. Because the flow is continuously present and the difference in the substance between these two dyes, there will be different in the time of separation. And so these two samples or dyes or material will separate at different time as shown in this diagram. The more volatile material are carried through the column more rapidly than the less volatile material, which result in the separation idea. This is a more animated show to explain it better. Here is the sample, and here we inject it in the column. Because of the difference in the passage of the different component of this sample, they will be separated and detected by a detector here. Each time the sample passes, the detector will detect it, and a graph will be shown on the graph side of the apparatus. A more descriptive diagram, this is the gas present in a cylinder. This is inert, of course, to the sample injected and to the stationary phase. This here is the coil containing the stationary phase. Here we inject the sample, and the carrying or the mobile phase will continue flow through the coil, and at the end a detector is present to detect the different substance that is separated in the end. All of these are present in a thermostatic oven to fix the temperature of this process, because temperature is very important for the accuracy of such a process. Now here is the picture of the apparatus itself and its different component. Again, the carrying gas is present and the sample size is injected through this injector port, and this is the column. Then after the material or the sample is separated to its different component, the detector here will write the graph intended on the computer side of the machine. Now, the detector could be either a flame ionizing detector, a thermal conductive detector, which is called the cathometer, or the electron capture detector. I'm going to give one example, the flame ionizing or ionization detector. How does it work? By the sample, when it reaches the flame will ionize this material or the sample injected, and this ionization will result in a graph formation after it is detected in a sort of electronic or electric signal. Now, a very important thing to know is the TR time. The TR is the retention time, which is the time between the injection and the peak of the signal. The center of the injection, or the zero, and the highest point or the peak of the triangle or the curved drawing. Each material or substance has its own TR time or retention time. By knowing this retention time for different material, we can know the material, how it is made of, or the component of the material. Now, we should know very important thing. Before we inject the sample, we should have some idea about the component of this sample. And we should have a control or a calibrating sample with us to know the TR or the retention time and the amplitude of each material. And by comparing the control sample to the graph that we have from our sample, we can know the component and we can know the amount of each component. The amount of each component corresponds to the height or the magnitude of the signal that results from this sample, as we can see here. And this time represent the kind of the sample or the component separated, the type, if you can say that. This is like a more realistic diagram that we can give as an example, and we can see the different materials present in our sample with its different curves. Thank you. I hope you enjoyed the lectures.